fall of 21, and she's already graduated. She's very young, but very accomplished also. She did a lot of work doing research with me. Somewhere in the middle of our research, we had to change our topic, and so she had to get a bunch of results in a very short time because her one credit hour was off. So she used a lot of extra time, and I want to appreciate her for that. So.
Thank you. So as Dr. Fidel said, my name is Westphal, and today I'm going to be talking about how prolactin receptor expression in meat uterus, ovary, and immortalized epithelial cells. So really quickly, just a brief overview of the entirety of this research that Dr. Fidel has been working on. It's more about the reproductive system and implantation. So I thought it would be smart to go briefly over the human reproductive cycle versus mink. So many of you guys are probably familiar with this, but it begins with fertilization. This is the sperm and the egg. They come together and form a zygote. After that, in 6 to 9 days, that is when implantation occurs. This is when that zygote is going to be implanting into the uterine wall of the endometrium. It's kind of the first trimester, second trimester, third trimester, parturition or childbirth. Pretty simple, pretty common knowledge. So in this experiment, we are using a model organism known as mink. So they go through a slightly different reproductive gestation cycle, and this occurs in 37 to 91 days in comparison to our around 9 months. So how is this different? So the first phase occurs around January or February. This is known as anestrus. So think of it as the pre-puberty stage. Then around late February to early March, we have estrus. This is when they are going through puberty. They are making the hormones that they need in order to eventually start mating. This is when fertilization occurs again. And one thing about fertilization is that this process is very successful. It has about a 90 to 100% success rate. So one thing that makes mink a great model organism is they go through something called embryonic diapause. And basically what happens is that this uterus kind of has a temporary arrest so that implantation doesn't occur for about 60 days or up to 60 days, whereas in humans we only have around 6 to 9 days. So that implantation will occur in late March to early April, and then they will also give birth. So prolactin is found to increase during embryonic diapause in mink. So as you can see, there are 3 peaks on this graph. I don't know how to do this slide. Oh, there we go. So estrogen or estradiol is highest during mating, induced ovulation, and then also in implantation. And then P4 or progesterone kind of peaks up after implantation. And we know that prolactin also kind of begins this peak during embryonic diapause. So our big question for this research is that we know that prolactin is there. We know that it starts increasing during this embryonic diapause phase. But what tissues specifically are targeted by prolactin during the reproductive cycle? So for this experiment specifically, we decided to look at mink uterus tissue, mink ovary tissue, immortalized epithelial cells, also known as GMME, and then estrogen-treated GMME cells. So here is like an overview of our research steps. So first, I actually created my own prolactin receptor primers, and then we isolated RNA from the tissues and converted that to C or copy DNA. Then we amplified the genome with PCR and looked at that gene expression through gel electrophoresis. So one thing before I created these primers, I had to figure out what made up a good primer. Generally, when you're in a research lab, you have pre-made primers for you, and this was a little different and something that I was very unfamiliar with. So doing some research, it's good to have a 40 to 60% GC content, and on top of that, it's also good to have the sequencing ending in a G or C because it promotes binding during that PCR stage. A melting temperature between 65 and 75 degrees Celsius is ideal. Balanced GC and GC domains. no runs of 4, so what this means is you don't want something like A, C, 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 lots of other variations of that. And then a product length of 300 to 500. This seemed to be the ideal range, so it wasn't too short, not too long. So I understand that the font here is pretty small, but these were the 2 prolactin primers that I ended up going ahead with. And just to look at some of their stats. So for primer pair 1, this one had about I want to say 52-53% GC content. Prolactin primer 2 had around 60%. If you look at the ends of the forward and reverse primers, the first 2 lines at the top, all 4 of those on the primer pair 1 and 2 end in G. And there was no runs. And then also if you look at the product length, the first one was 509, which was really close to the range that we wanted, while the second one was 499, which was in that range. So just as I continue along in this presentation, primer pair 1 is going to be referred to as PRL1, primer pair 2 is going to be PRL2. So continuing on, we did the RNA isolation and conversion to cDNA to prepare these tissues for gene expression. Basically, RNA isolation is when you are separating that pure RNA from DNA and other proteins. So we did this with the mink uterine tissue, mink ovarian tissue, and amortalized epithelial cells, the GMME cells. And for those of you who don't know, so the darker purple at the top, that is the luminal epithelial cells, and then the circular ones are going to be the glandular epithelial cells. And then one important note is that when we did finish this part of isolation, we had a purity ratio for the 260-280 at around 1.68 to 1.78 throughout all of our samples. A perfect rate is around 1.8, so the closer it is to that, the more accurate our results are. So then moving on to the conversion to cDNA. cDNA is copy DNA. The difference between regular DNA and cDNA is that the cDNA is only going to have sections of coding sequences, whereas regular DNA is going to have coding and non-coding sequences. So basically this is synthesized from our isolated RNA with an enzyme called reverse transcriptase. And basically, a little overview of how this works. So it's going to start with primer annealing. That enzyme is going to help build that second strand on. And then DNA polymerization is going to occur. And once that is done and it has become double-stranded, that enzyme, reverse transcriptase, is going to deactivate. Next, we did PCR and gel electrophoresis, which allowed us insight on our gene expression. So for our PCR, we did CNA mixed with DreamTat Green PCR master mix. What we did, we added the PRL1 and PRL2, both of the prolactin receptors mentioned before. These were our forward and reverse primers. And then we also used insulin receptor as a control during this process because previous research had indicated that this was already present in ovary and uterus tissue. And for those of you who are unfamiliar with this process, it basically is 3 main steps. So there's denaturing at 95 degrees. The strands are separating from one another. Then there's annealing at around 55 to 65 degrees. This is when those primers are going to bind on. And then there is extension at around 72 degrees. And this is basically amplifying that genome. And these 3 processes make up one cycle. We ran ours for about 40 cycles. So then we have our gel, and basically gel electrophoresis is going to separate the DNA based on their size and their charge. And we actually ran 2 gels. So our first one was just the uterus tissue. It's pretty hard to see on here.
The light. There we go.
Yeah. The lines are a little faint, but I will get into that in a second. So it is a little hard to see, but our controls are there. So we know that our results are prevalent. So for the uterus specifically, PRL1 was expressed while PRL2 was not. We did the ovary GMME and estrogen-treated GMME on a separate one. So we used the INSR ovarian tissue control again, and that was prevalent. And so if you look closely at the data, so this is all PRL1. So PRL1 was expressed in every tissue type. But if you look over here, PRL2 was only expressed in GMME cells and estrogen-treated GMME cells. One thing to note If you're familiar with gel electrophoresis, generally the thicker and the bolder the band, the more that gene has been expressed. So we came up with a couple of hypotheses as to why our lines were pretty faint. For one, methods. It was my first time making a gel, so there definitely could have been some user error in that. Also, we ran two gels at two different temperatures. So the one with just the uterus was run at about 120 to 125 degrees Celsius, whereas the second one was ran at about 105 degrees Celsius. And so like, I don't know if you can tell, but this is our ladder, which is basically allowing us to see the expressions. This one's a lot bolder, so that higher temperature could have caused the fainter lines in the beginning. Second of all, we were using anestrus tissue. If you remember from the beginning of the presentation, this is the pre-puberty tissue. So prolactin itself isn't expressed as much normally in that. So looking at the female reproductive system, estrogen and progesterone, E2 and P4, are produced by the ovaries. And then these are, the uterus has receptors of the uterine wall, which is where estrogen and progesterone go into. Prolactin, on the other hand, is produced by the anterior lobe of the pituitary gland, which is right underneath the hypothalamus in the brain. So based on our data, it is proposed that prolactin is expressed in the uterus and the ovaries based on the estrogen and progesterone receptors found in the uterine wall. So as I said before, expression of prolactin receptors are found in uterus tissue, ovary tissue, GMME, and estrogen-treated GMME. However, because GMME and estrogen-treated GMME were the only ones that had both showing gene expression, we hypothesized that they are a potentially good model to prolactin's role in reproductive physiology and would be the best cells to use for future research. Also, just adding on that during estrus, PRL levels are present, but they're much lower than what they are once those mammals have gone through puberty. So this is something we kind of hypothesized for future experiments, prolactin playing an important regulatory role in mammalian reproduction and early pregnancy. So because we know that prolactin receptors are present in the uterus and they connect with the estrogen and progesterone receptors, we hypothesize that there is the potential for the production of nutrients, specifically carbohydrates, such as glycogen and glucose. And if that's the case, then prolactin could have some kind of regulatory role for successful implantation in the reproductive cycle. For future research, I think it would be very beneficial to go in with qPCR, quantitative PCR, instead of the PCR we did with the DreamTac. This would be using cybergreen, a fluorescent dye. So instead of just saying if the genes are expressed, it would be letting us know what amount the genes are being expressed in. And additionally, it would be very beneficial to look at immortalized stromal cells, which are GMMS cells. In the uterus, there are epithelial and stromal cells. So just looking at the epithelial gives us slight information on this, but in order to have a full and successful picture, it's really important to be looking at the stromal and the epithelial cells. And just for reference from that previous picture, the lighter purple is all stromal. And then understanding reproductive hormones is beneficial in many aspects of life, not only just for improving and understanding fertility like we're doing in these experiments, but more broad picture. We can help increase litter sizes, which in turn helps conservation of animals. Also, it can be very helpful in birth control and understanding more effective methods of this, as well as diseases and syndromes. And then I just want to give a special thank you to Northwest Nazarene University for giving me this opportunity, for letting me use the materials in the lab, for funding me. A big, big thank you to Dr. Hudanu for everything he has done for me, coming in on weekends, answering all my questions. And then I would also like to acknowledge Dylan Brewer for his help with cell culturing, and AJ for his help with RNA isolation and conversion to cDNA. Thank you. Are there any questions?
Time for one. Okay, then I have a question about the temperature. What were you, oh, so go ahead, Dr. Cassell.
Okay.
No, I'll ask it later. Go ahead.
I was just curious to know if there weren't any prolactin primers available, and that's why you had to create your own, or you just wanted to try that?
So there were prolactin primers available. It was actually Dr. Cardono's idea that he thought it would